Okay. Just as a recap, so I have it on there for the thing. We're going through Hello, people in the computer. We're going through cellular energetics. We can break this down into catabolism to three groups: digestion, breaking down macromolecules into simpler monomers, oxidation of those monomers to acetyl CoA, which is like a hub of all your metabolism. And then we take that acetyl CoA and we oxidize it and get out water, CO2, and ATP. Goodbye, people in the computer. That will be you someday. Okay. So let's really briefly talk about kind of overall how this happens. And the first process of this is this thing called glycolysis. Now, I'm going to go ahead and post up this individual figure, onto D2L, and I will do this fairly quickly. But we're going to talk more about really what's in here. I don't necessarily want you right now to like write this entire thing down. I'll talk about specific parts of this as we go along, so maybe like make notes on those, like about those specific parts of it, and then you can go grab the whole thing and talk about that in just a few minutes. But before we do that, we're going to talk about the quiz that you will have over this. That's going to be online again, like the previous ones. And I'll say a couple things here about this. It will go live Thursday at 8 a.m. So tomorrow at 8 a.m. this quiz will go live. You will have until Friday at 4 p.m. to finish it. So that is the time frame. Now, if you don't get it done in that time. If you don't, I will this time not likely be inclined to extend that a little bit. So, Friday at 4 p.m. On this one previously, I had 30 minutes for the previous one. I think I'm going to make this one the same, so you'll have 30 minutes again to go through this. And I'm going to ask you about intermediates and the enzymes that are between places, where ATP is formed, where ATP is used, when NADH is formed and used. We will talk about this as we go on. And then I'll have a couple questions also in there that are conceptual. For instance, imagine this happens. I really like imagine the toxin kills this. What would happen?
It dies.
Yeah, things die, but like very specifics about like how would that work. It kind of tests if you understand like the concept of how things are working to understand how things would break if something happens. So, will you have that? Are there any questions on that quiz? Everyone kind of have the whole online thing figured out? Okay. Okay. So let's talk now about glycolysis, and let's talk about some major points in glycolysis and what we're doing. So, first off, one major initial important point here is that, number one, you're taking glucose. Glucose is what you were putting into glycolysis. The thing that you get out is pyruvate. Glucose is a 6-carbon molecule. You'll remember that's a hexose. We have this 6-carbon sugar. And what we're going to get out is 2 of these 3-carbon molecules. So just to kind of illustrate how that goes here, we have glucose here. We are going to run it through several intermediates. So glucose, we're going to run through several intermediates, which I'll talk more about later. At this point right here, where we have fructose bisphosphate aldolase, right here, this breaks fructose, 1,6-bisphosphate, into 2 glyceraldehyde, into 1 glyceraldehyde 3-phosphate and 1 dehydroxyacetone phosphate. And then we use triose phosphate isomerase to turn this dehydroxyacetone phosphate into a glyceraldehyde 3-phosphate. But through the big picture here is that we take this fructose 1,6-bisphosphate, and through the action of these 2 enzymes, we get out 2 glyceraldehyde 3-phosphate. And so that's the point in which this reaction, we get 2 molecules out of this. We get 2 3-carbon molecules out of this. from one 6-carbon molecule up here. And then we run the rest of this reaction through here, and we get out pyruvate at the end. So again, just kind of summing this up, you have glucose, a 6-carbon molecule. We go through some reactions. One of those intermediates, fructose 1,6-bisphosphate, gets turned into 2-glyceraldehyde 6-phosphate into the action of 2 enzymes. And then eventually we get out, through some more intermediates and more steps, to pyruvate. Another important point of this is glycolysis is happening in the cytosol of the cell. We're going to be talking later about specific other portions of metabolic pathways that are happening in specific organelles, specifically talking about Krebs cycle, or the citric acid cycle, and electron transport chain, which happen in the mitochondria. But this doesn't. Glycolysis does not happen in the mitochondria. This is happening just out loose in the cytosol. That will become important as we go along. Also, we are using 2 ATP at the beginning of this process. The whole point of everything that we're doing is to make ATP, but to get the entire thing going, we are using 2 ATP. And we're taking one of those phosphates and sticking it onto glucose to make glucose 6-phosphate here. So we stick a phosphate using ATP. So this actually uses up 2 ATP because we need an ATP to actually run it, and then we actually need one to get back. But we're using 2 AT at the beginning. Actually, hold on, not that one. Scratch that. We use 1 ATP here to put that on here. Then we use another ATP here to stick on the other phosphate on there. So 1 ATP, 2 ATP used. And this is really important, especially this first step. This first step is going to be catalyzed by hexokinase, is going to take, as its name says, is going to stick a phosphate onto a hexose. And what this does is this is kind of slick. We actually take glucose, we stick a phosphate on it. It's now glucose 6-phosphate. It's not glucose anymore. Now this might seem like a technicality, but it actually works this way. It's not glucose anymore, which means that because we are only letting glucose into the cell through diffusion, we need to have a high concentration gradient of glucose inside the cell to outside the cell to actually run this diffusion. If you just start building up glucose on the inside of the cell, the rate of glucose diffusing into the cell is going to slow down. So, if we put a phosphate on it, it's not glucose anymore, which means that there's low glucose actually inside the cell. No matter how much you get in, you just keep sticking phosphates on it, it's not glucose anymore. There's a low glucose concentration inside the cell, which allows for the continuation of diffusion of glucose into the cell. It kind of seems like what brought on the financial crisis. It sounds like cooking the books almost. Like, this isn't glucose. It has a phosphate on it now. Don't worry about that. Keep giving us more glucose. Like, this really isn't money. Keep giving me more money. But it actually works this way. So you stick a phosphate on, it's not glucose anymore. You can just keep loading up the cell with glucose because, hey, I don't got any glucose. Another thing that it does is it traps the glucose on the inside of the cell. So glucose moves in, and it goes through a glucose transporter, and as we were talking about this carrier-mediated diffusion, so it's going to bind, it's going to go through, that's all grand. But once it's in, you stick a phosphate on it, it can't go back out the other way because, again, it's not glucose anymore, it's glucose 6-phosphate. And so these two things allow you just to keep moving glucose into the cell. Now, all that being said, it's like you're pumping it in, and in truth, you kind of are. This takes energy. This is going to take an ATP, so it's not like a totally free, completely cooking the books. It's like you're still paying taxes on that money, so to speak. It just looks shady. I've never heard, I'm proud of myself, I've never heard anyone talk about biomolecular processes looking shady before, but okay. Anyhow, so I think this is slick. I like how this works. So again, that's right up here. We're attaching an ATP to glucose, get this glucose 6-phosphate, and voila, it's not glucose anymore. We made it disappear. And then again down here, we stick another glucose or another phosphate on, because if we split it and we want it to be symmetrical, like we get the two things out of it, we need 2 phosphates on it. So we are investing 2 ATP to begin this process. Okay. So, and that's mediated again by the enzyme hexokinase. This is involved in the phosphorylation of that glucose. And it prevents the glucose from exiting the cell, and it lowers the intracellular glucose concentration. That's the dry way of saying it. That's just kind of saying it does these things. But to me, this is like so cool. This makes me happy. It makes me feel like all my cells are used car salesmen in a way. Never mind all of that stuff there, all that rust. That's not really rust. It's special. Okay. Another important point here is that glycolysis is going to generate 4 ATP. That's kind of nice that it generates more ATP than it uses. But we'll generate 4 ATP during that process. And we're doing this through something that's called substrate phosphorylation. So we're actually pulling, instead of actually using just the phosphates, we're actually pulling the phosphates off of some other substrate and sticking it on ATP. I'll talk about what we're talking about here, or show you what we're talking about here. So we're actually pulling that phosphate from another molecule. So for a moment, let's take a look at what we're talking about there. So you'll notice down here we have another phosphorylation step right here, where we take a phosphate and stick it onto ATP from 1,3-bisphosphoglycerate, and then over here, that's being phosphoglycerate kinase. And then up here, pyruvate kinase is also going to take another phosphate and stick it onto an ATP. So we have 2 more steps in which we have ATP being generated. Now that being said, remember, by this point, after this point right here, all of this is doubled because we have now made 2 3-carbon molecules. So that means, actually, for every glucose that we stuck into here, we are getting 2 ATP made at this step, and we're getting 2 ATP made at this step up here. So we actually have a couple of them. Yes, what's that?
What does the magnesium, the magnesium 2 plus provide for the enzyme? Is it just helping the enzyme?
It is, I believe in this case, this is a cofactor that's being used. Yeah. Good question. So we have that, and then we also form 2 NADH. NADH stands for Nicotinamide Adenine Dinucleotide. Which is something derived from niacin, which is a vitamin. And for the moment, you won't see the, like, you have a good idea what NA or ATP does for you. It's going to power all these reactions. But at the moment, you're probably looking at NADH and being like, so what? It makes alphabet soup up there. I like that, but not since I was 5, so. Let's look at where it does this just for a second here. We look over here, and we're getting 2 NADH out right here. We're actually making it from NAD plus and turning it into NADH by adding a hydrogen onto there. And this is what NADH looks like. We have adenine right here. We have nicotinamide up here, and then it is just 2 nucleotides. So a nucleotide of adenine, nicotinamide nucleotide, and then just like you would use the same sort of bond here that you would make DNA, we have, or RNA, depending, I guess this is oxy, so this would be the same as a RNA, but you have the same sort of phosphate backbone, but we only have 2 nucleotides, and one of them is not like a canonical, like DNA, RNA nucleotide. Or to put this in a little bit easier way rather than that, we have nicotinamide, ribose phosphate, we have an adenine, ribose phosphate. Those are connected together through their phosphates. And on the nicotinamide, we can stick an extra hydrogen, an extra proton, which gives us what we are going to be talking about later on. Now, this is going to be important again in electron transport chain. This is really, the NADHs are what are going to drive electron transport chain. Because we're going to get electron donation from this bond right here, and then run that thing. But for now, just kind of store this away. We're getting 2 NADH made for every glucose that we put into glycolysis. So this NADH is a coenzyme and a mobile electron carrier. Each NADH is going to carry 1 proton and a pair of electrons. Oh, sorry. I'll try not to keep running together. Are there any questions at this point on glycolysis? Okay. We're now going to go into Krebs cycle. Now, next week, the quiz will be over Krebs cycle and probably electron transport chain, depending on how much of the electron transport chain we're going to get through. And again, it's going to be kind of similar. What are the names of the intermediates and enzymes? Where is NADH formed and used? Where is CO2 produced? And what kind of involvement do you have of acetyl CoA and FADH? Actually, not acetyl CoA, just coenzyme A and FADH, which we'll talk about here in a moment. And again, there'll probably be, like before, several multiple choice, and then maybe some short answer, and then a couple kind of larger, more conceptual questions about understanding the overarching process. So let's take a brief look at what's happening here during the citric acid cycle, or Krebs cycle. So we have that pyruvate. This pyruvate right here has come from glycolysis. We now have that out. And we take a coenzyme A here and react it here and get an acetyl CoA out of that, which then feeds into this. So now what we've done here, we are taking a 3-carbon molecule and feeding it into this cycle. In doing that, we're losing 1 CO2 right here. So our net, at this point, number of carbons that we are putting into this is going to be 2 novel carbons. Does that make sense? We have 2 carbons left from each pyruvate, but that means we actually have 4 from each glucose. Also, at this step of pyruvate dehydrogenase, we actually run through and get out a NADH. So we produced another NADH at this step as well. This acetyl CoA then is going to then react through citrate synthase and oxaloacetate to get citrate. And now we're back up to, so this here, oxaloacetate, is a 3-carbon molecule citrate plus this. Now we have a 4-carbon molecule. Hold on. 6-carbon molecule. Here. Yeah, 6. So it's a 4-carbon molecule to a 6-carbon molecule. We got a 6-carbon molecule, and then, here we go. It's even illustrated right here. Before, that was the oxaloacetate right here. Here we have the acetyl CoA right there that has been added onto that. And then through several reactions, you're going to get this deisocitrate, which we're going to go through isocitrate dehydrogenase and produce another NADH. At the same time, we're also going to produce a carbon dioxide at this point. So we're cleaving off another CO2. And then we come down and we get through that, we arrive at alpha ketoglutarate. And going through alpha ketoglutarate dehydrogenase, we get succinyl-CoA. And also, again, through this process, getting another NADH produced and another CO2 produced. Okay, at this point, we have now used up those two novel carbons that we have introduced. Those have both been used. And so the rest of this, conceptually what we are doing, is going through and essentially regenerating the citrate. We do get a couple additional things. We are going to make, from succinyl-CoA synthetase, we are going to get a GTP produced. That is going to be important later on. We also here, from the step from succinate to fumarate, from succinate dehydrogenase, we go from Q to QH. This is actually forming an FADH right here at this step. So we get another electron carrier produced at that step. And up here again at malate dehydrogenase, which converts malate back to oxaloacetate again, we get another NADH produced. And then we return back up here to oxaloacetate through citrate synthase, gives us citrate again, which we're going to use to add, or has been added the additional acetyl-CoA, and then we can run around the process again. So, here is a couple conceptual things that I want you to look at here. Okay, I'll actually come back to those in just a moment. Okay, another way of looking at it, again, both of these, I'm going to put on these figures onto D2L. We start off with a 3-carbon molecule, lose a carbon off that, and get acetyl-CoA. We add that to oxaloacetate, which is a 4-carbon molecule, get a 6-carbon molecule. Run through in the step from isocitrate to alpha ketoglutarate, we're losing a CO2 and get a 5-carbon molecule, and then the next step, alpha ketoglutarate to succinyl-CoA, do alpha ketoglutarate dehydrogenase, we're getting another CO2 loss, another NADH performed, or another NADH created, but now we're back down to a 4-carbon molecule, and through here we're generating another GTP, which is going to be important. Succinate to fumarate, we're getting another electron carrier formed, FADH2, and then back up here, malate to oxaloacetate, we're getting another NADH created. So this now, as opposed to what we were talking about before, this is happening in the mitochondria. Not happening in the cytosol. So that means that the pyruvate that we generated and the oxaloacetate, in those steps we are transporting this from the cytosol into the mitochondria. Talk about that a little bit more later. In general, what is happening here, so we get pyruvate, we're losing a CO2, and that gives us this 2-carbon molecule, oxaloacetate, or acetyl-CoA, excuse me, which then we add on this 4-carbon molecule, and then this, was supposed to be over here. My format got screwed up when I projected this, apparently. So this is supposed to be here, but we get this 2-carbon plus this 4-carbon molecule, oxaloacetate, get 6-carbon molecule, citrate. And so we go through a cycle in which we lose 2 CO2, generate 3 NADH, generate 1 FAD2+, and generate a GTP, and get back to oxaloacetate, and losing the 2 CO2, essentially losing those 2 carbons along the path. So yeah, this, forget about that for a second. Put that over there. That will be fixed on the slides I put up on D2L. So that is so crazy. another point is every time you put one glucose into glycolysis, you're going to get out 2 pyruvate, which means that you're going to run through this process twice around. You're going to run through Krebs cycle twice, or once per acetyl CoA. We are also going to generate 3 CO2 per cycle, but 6 per glucose. Now this should intuitively make sense. For every glucose that we put in, we have a 6-carbon molecule. We are taking and essentially burning it up, completely using up the energy to it, so we have a really low entropy state in glucose, and we run it to relatively high entropy molecule CO2 that you can't do much with. And in doing so, converting this glucose to CO2, we are capturing the energy that was in that. We are going to produce 4 NADH per cycle. Again, one when we took pyruvate and turned it into oxaloacetate. Again, we take isocitrate and make it to alpha-ketoglutarate. Again, when we take alpha-ketoglutarate and turn it into succinyl-CoA. Again, we take succinate and malate and turn it into oxaloacetate. We're going to produce 1 FADH for each of these cycles, or again, 2 for every glucose that we put into it. So let's talk just for a moment about what is FADH. We talked about what NADH was. Let's talk about what FADH is. So this is flavin adenine dinucleotide, so making this similar to NADH. And this is a derivative of riboflavin, which is vitamin B2. And to conceptually give you an idea of what this would look like, that second flavin down here should be an adenine. Sorry about that. So you have a flavin, you have now 2 hydrogens attached to that, plus electrons connected to a ribose, to a phosphate. So again, we have 1 nucleotide here, and we have an adenine right here that is like trying to pretend it's a flavin, so sorry about that. But you can see through that in disguise. I know you can. Connected to a ribose and connected to a phosphate, so again, a complete nucleotide, and those 2 nucleotides connect together to that phosphate bond there. And this is a prosthetic group. This is going to be bound tightly to an enzyme. We're going to talk more about this in a little bit of detail later, because this enzyme is actually important in electron transport chain. We're going to take a little bit more time and go a little bit more carefully through electron transport chain, but this is connected to that enzyme and is important to its function. And this is like NADH, this is an electron carrier, carrying 2 electrons and 2 protons that it can donate. So we're also creating 2 GTP for every cycle that we go, or every glucose that we put in, once for every cycle. That will be important as well. This is very similar to ATP, but instead of being adenine triphosphate, we have guanidine triphosphate. Again, just simply swapping out the nucleotide, adenine for guanine at the phosphates are connected to. And we can actually use this to go back and create ATP at later time. So we can take a GTP plus an ATP, take a phosphate off the GTP, put it on the ADP, and we get GDP, guanidine diphosphate plus adenosine triphosphate. Yes, Alex.
Why don't we just make ATP from ADP originally instead of making GTP? And what's the reason we're going to choose to use guanidine instead of...
You know, to tell you the honest truth, I am not exactly sure conceptually why that happens. Yeah, that's interesting. I'm now curious about that. If I figure that out, I'll let you know. Yes, Dave.
So is this saying that all of the GTP produced is going to be turned into ATP, or just some of it?
Virtually most of it. So here's the thing. How physiology and a lot of other things work is when you actually do this, GTP is going to go into a pool of GTP that is existing in the mitochondria, which then gets used for various things, right? Now that being said, I believe the molar amount that you produce of GTP plus the molar amount of GTP that you use to make ATP is similar. But that being said, saying that's molecule for molecule the exact one that you're using would not be accurate. Does that make sense? Yeah. Because you're using, the mitochondria uses GTP for other things as well. However, that being said, conceptually you can think of it as it's all going for that. But in reality, that's not the case. Does that make sense?
It's not just being like carried to somewhere, it could be turned into ATP,
No.
lickety split.
It is not. And again, that's happening here between succinyl-CoA and succinate. Actually, with that, we're going to start talking about the mitochondria. So with that, I will start handing back tests. Take a sack. I will take a sack. We will start going. I'm going to stop.